So continuation of regenerative endodontics. Next one is platelet-derived growth factor. Platelet-derived growth factor is released by platelets and has potency in promoting angiogenesis and cell proliferation. In trauma, hemorrhage is followed by blood clot formation in dental pulp. Platelets in the blood clot release a granules containing PDGFs and attract neutrophils and macrophages. So after injury, platelets is converted to activated platelets and which in turn release growth factors. Transforming growth factor beta. The transforming growth factor beta family comprises a group of diverse growth factors including TGF beta, bone morphogenic proteins, growth or undifferentiated factors, an anti-Mullerian hormone, activin, etc. TGF beta 1 also plays an important role in immune response during dental pulp injury, helps in healing. Bone morphogenetic protein. Bone morphogenetic proteins comprise a subgroup of TGF-beta superfamily and are involved in many biological activities, including cell proliferation, differentiation, and apoptosis. BMBs have strong osteoinductive and chondrogenic effects. Vascular endothelial growth factor. Vascular endothelial growth factor is a heparin binding protein with specific affinity to endothelial cells and plays a key role in angiogenesis. The functions of vascular endothelial growth factor involve the proliferation of endothelial cells and their enhanced survival, stimulating neovascularization in the area of injury. Vascular endothelial growth factor appears to induce the differentiation of human dental pulp cells into endothelial cells. Fibroblast growth factor. Fibroblast growth factor plays key roles in cell migration, proliferation, and differentiation during embryonic development and wound healing. Insulin-like growth factor. Insulin-like growth factor are single-chain polypeptides that have a high sequence similarity to proinsulin. Insulin-like growth factors comprising IGF-1 and IGF-2 contribute to odontogenesis and dental tissue repair by cell proliferation and differentiation. Nerve growth factor. Nerve growth factors, also known as neutrophins, promote the survival and maintenance of sympathetic and sensory neurons. Nerve growth factors induce the differentiation of immortalized dental papilla cells into odontoblastos in vitro, suggesting that nerve growth factor acts as a stimulant for mineralization. Scaffolds. Scaffolds is a three-dimensional structure that contains growth factors. The role of the scaffold is to provide support for delivering cells and or growth factors to the proposed site of tissue regeneration. There are important features to consider in scaffold selection, including the physical and the mechanical aspects of material and its biocompatibility and its degradation time. So classification based on the degradability of the matrices, biodegradable scaffolds, permanent scaffolds, based on form, solid blocks, sheets, porous sponges, and hydrogels, based on the presence or absence of the cells, cell-free scaffolds and scaffolds seeded with stem cells. Based on origin, biological or natural scaffold, platelet-rich plasma, platelet-rich fibrin, collagen, chitosan. Artificial or synthetic scaffolds, polylactic acid, polyglycolic acid, and polyepsilon caprolactone. These physical aspects of 3D scaffold include porosity, pore volume fraction of the scaffold, pore size, pore diameter, and pore structure, that is the shape. Important mechanical properties of scaffold material include viscoelasticity and tensile strength. For dental regeneration purposes, the tensile strength may be not as important as viscoelastic properties of the scaffold materials. A beneficial clinical feature for dental pulp regeneration would be if the scaffold is injectable, as are some of the natural scaffold materials and hydrogels. In these cases, the gelation time would need to be taken into consideration when seeding cells in a scaffold for implantation into a host. An essential clinical feature for scaffold selection is biocompatibility. Naturally derived scaffold materials have the advantages that they are generally well tolerated, do not lead to immunogenic response and do not involve the use of harsh chemicals during processing. However, a major drawback is the lack of control over pore size and heterogeneity of the scaffold. Degradation process of the scaffold is important and should closely follow the rate of tissue regeneration. Ideal requirements of a scaffold include should be porous to allow placement of the cells and growth factors, should allow effective transport of nutrients, oxygen, and waste, should be biodegradable, leaving no toxic byproducts, should be replaced by regenerative tissue while retaining the shape and form of final tissue structure, should be biocompatible, should have adequate physical and mechanical strength.